Hello, my name is Jackson Harrell, and today my bioseminar topic is on the effect of transgenic Bt corn on agricultural animals. So basically, the reason that I chose this is because I wanted to pick something that was seen as relevant in our world as humans, and genetically modified foods are a big topic. It has a lot of controversy surrounding it, and that's why I chose the topic in the end. And here's the outline. We're going to talk about how GMOs are made, what Bt corn is, uh, why we care about those effects on agricultural animals. I'm also going to talk about the experiments, two experiments to be exact, one short term and one long term. And then finally, a few take home messages. So GMOs are genetically modified organisms, and they get this bad rap because of just that. People that are uneducated in this topic see genetically modified foods and instantly, think that it's going to have a negative effect on our body, and in the end, have a negative effect on our lives. But in reality, genetically modified organisms are here to help us. Um, there's an article online from Forbes magazine. Obviously, not a scientifically credited magazine, but I see this as kind of like the mid between, something that has no factual information and something that is purely scientific. A lot of Americans kind of see this writing as something that's informational for them. And Forbes magazine came out with a article that said that basically genetically modified foods get a bad rap because of the process within it. And that process pretty much you can see down here in the bottom left. The process basically goes from they take the DNA strands from these crops and they splice out with enzymes, what DNA strand that they want to affect. And basically, they're cultured in bacteria in order to get the strands that they want. And then, in order to place it back inside the host, they use a virus to put it inside the host cell or in the host nucleus. And then from there, the DNA develops and begins to divide and affects the whole plant. And then the plant grows the entire crop with the genetically modified fruit. So basically, they've said that GMOs get a bad rap because of that process. Just hearing bacteria and virus being used for foods that people have known for hundreds and hundreds of years and have grown, and now people are seeing that they're trying to change that, and they think that brings a bad rap. Actually, the first genetically modified food was the Flavor Saver tomato. This was created in 1994. And basically, they did this in order to create a longer shelf life due to being able to delay the ripening process of those tomatoes, making it easier, to sell, to ship out, and basically have industrialized manufacturing for these tomatoes without having them, you know, be bad and old within, a couple of days of transferring. So what is Bt corn? Bt corn, the Bt stands for Bacillus thuringiensis. It's a genetically modified corn that produces a toxin called Cry1AB. And basically, this toxin enables for the resistance of crop pests and other toxins. One of those crop pests is the European corn burr. You can see down here in the bottom left, basically makes the corn stalk itself as a host. It just feeds off of that and lays its eggs there and basically deteriorates the whole nutritional compound of the corn. And so on the bottom right, you see the difference between Bt corn and non-Bt corn. So Bt corn is transgenic because of the introduction of new genetics and the Isogenic corn is the corn that has grown naturally without any genetic modification to it. So obviously you see in this picture down here, BT corn is a lot more resistant than to mold than the non-BT is. So why is transgenic corn better? Fumoniacin B1 is a toxin that affects corn, and basically what the BT corn does is it hinders and eliminates the amount of fumoniacin in that corn. So obviously mold creates a less nutritional corn containing myotoxins that affect our body negatively. You see right here is the chemical compound of fumoniacin. And then down here, obviously you have some corn with mold on it, and that those mycotoxins can affect all kinds of parts of a farm animal. You know, you see in this pig, it creates, infertility, um, decreased litter size, smaller birth weight, liver damage, all kinds of stuff. So it affects the body in many ways. So with genetically modified foods, more specifically the BT corn, it can help eliminate these problems. Why does this matter to us? Looking at short and long-term effects of animals can be used as model systems to show how these genetically modified foods affect us as well as affect the food that we eat. So you see down here is some pigs, and then you see some bacon over here, which I don't know a lot of people that don't like bacon. So the first experiment is a short-term experiment. It looked at the growth of weaned piglets. Weaned piglets are in the transitional state from milking from their mother to solid food. And so basically, this study looked at the growth of these piglets over 35 full days in the most crucial growth and development stages of a pig. So the purpose of this experiment was to compare the effect of BT corn and isogenic corn on the growth of these weaned piglets. So basically, they took 128 weaned piglets weighing around 9 kilograms of live weight. They were randomly assigned to 4 groups of 32 animals each, 16 males and 16 females. So this experimental design was performed in Italy. They basically took 2 farms and split it up between isogenic and BT. All the piglets were picked that had no previous health issues, because they didn't want to ruin their experiment by, using piglets that already had health conditions that would hinder them. So 32 pigs were fed isogenic corn and 32 pigs were fed the BT corn at the farms. So you see down here in this little graphic, 2 farms, you have 16 males and 16 females being fed isogenic, and you also have 16 males and 16 females being fed the BT corn. And they ran 2 duplicates of this in order to get better results. So here's the main results of this experiment. You see here in table 2, what this arrow is pointing at is the fumoniacin B1 concentration. You see here you have 2,500 to 378 for the BT corn, and then you have over 5,500 micrograms per kilogram of dry matter, and here you have 2,000, which is a lot smaller. And then this table is also showing everything else is basically the exact same, whether it's crude protein percentage, starch percentage, digestive energy percentage, all that really shows that these corns are very identical, except for those one tiny little changes that can create nutritional benefits for the long run. So you see here in table 4, this shows the average daily gain of these piglets as well as the feed intake. And then they also combine that to see the feed to gain intake. So what I want you to get from table 4 down here is from basically 0 to 14 days doesn't change that much. You see 15 to 35 days, another change that doesn't really change all that much. Oh. And when you look at the feed intake, you see a little bit lower feed intake with the BT corn as compared to the isogenic corn. And then the feed to gain intake is where it really takes off. And this feed to gain intake is basically seen in this graph right here. So this graph is showing both the males and females feed intake. And you see in table 4, basically the average daily gain is about the same. But if you go over here and look at this table, this graph, the feed intake for the BT corn is way less. So basically what that means is these pigs are getting more nutrition and eating less food. So this is good for both the pigs as well as from a economical standpoint. You have these farmers that are not having to give them as much feed while they're gaining the same amount of weight. So the second experiment is a long-term experiment, and what they did was it was a 10-generation study where BT corn was used as food for quail, and they looked at the weights just like the pigs, but more importantly, they looked at the fertility and survivorship rate of the chicks. This was more of how it would affect these chicks that were, 5 generations in, 9 generations into eating this BT corn, seeing the effects of that, and seeing how that affects the hatchability and fertility rates of these animals. Down here, you see a mother quail with all her little chicks. So the purpose of this was to test the long-term effects of BT corn on quails and compare those results to the isogenic corn. So basically, the experimental design here, this was performed at a lab in Japan. It was around 70 males and 75 females per generation. Half of them were fed isogenic, while the other half were fed BT corn. And 210 eggs were collected per group in each generation. So down here, another little schematic of the process. You see here you have 32 females, 30 males being fed isogenic corn, 32 females, 30 males being fed the transgenic corn. And basically they ensured that these eggs would be produced by having more females than males in every single situation to enable this kind of fertility. So this, after they collected the 210 eggs from them being from them after laying them, they analyzed the results of the hatchability as well as what they did was when those parents of that generation were done reproducing those eggs, what they would do is they would kill them, and then they would look inside their gastrointestinal tract and look for changes in the DNA of the gut and the stomach in order to see if the BT corn had changed their DNA in some way, or if it had changed, the egg's DNA in some way. So here we are at another table. This is the two most crucial graphs. The first graph is the laying intensity percentage. So basically, they wanted to look at, how often these quails were laying eggs, as well as how the number of eggs were. And so in this table, you see that the laying intensity doesn't really change much, stays about the same. So through this 10 generation period, you see fluxes, but none of it's really statistically significant. And same thing with this graph. This is the hatchability of eggs. Obviously, the 210 eggs, all of those eggs aren't going to make it. That's just the way that nature works out. And um, you see here that it's basically the same. One thing interesting to me was that even though it's not statistically significant, you see here at the beginning of the generation, the isogenic corn, which is labeled here by the solid lines, um, is is higher, basically all the way through until the end, and then you see kind of a change, and that was really interesting to me. But obviously, it's not statistically significant. Um, the transgenic corn, as you can see with the striped bars here, they kind of has this. They're all about the same, but like I said, at the end, it kind of has a little bit of an uptick, but not enough to be statistically significant. But they're not trying to prove. This study is not really trying to prove that um, BT corn is better. This study is to see the long-term effects of you know how how this affects fertility and survivorship of these animals because of how long their family line has been eating this BT corn. And the stigma around it is that since it's genetically modified, and you have to go through this long process of enabling transgenic corn. Some people out there believe that it's bad. It creates a reduction in infertility, but it doesn't. That's just not true. So future studies, model organisms shed light on the aspect of GMOs on the body. Obviously, we saw with the pig that it showed, less feed intake, but the same amount of gains, if not more. And then, with the quails, you see that they, um, the hatchability and survivorship of these animals doesn't really change. It stays almost constant all the way through. Future tests could be conducted for humans in generational studies. Obviously, that wasn't the point of this bioseminar topic, but you know, people want to know how it's going to affect them and whether or not they should be eating it. But a long-term generational study would really help in clarifying those people that think that GMOs are bad. A couple take-home messages. BT corn and diets for these model organisms do not have any detrimental effect, and they can improve performance and growth due to the lower human biotin 1 B1 levels. Um, in the long-term generational study, they had no graphic to determine this, but they had this all over the results. The DNA analysis in the quail right after feeding showed that transgenic DNA was in the gastrointestinal tract, but 12 hours after, DNA was untraceable in the quail. And this was interesting to me because, that whole study is based on how that DNA is going to affect the body of us. But obviously here, you see that there's traces in it, but, after 12 hours, all of that's gone. This shows that BT has no harsh effect on animals that consume it. And I think that, that goes a long way. And here we go with a nice little picture of a cornfield. I think that, these studies really do shed light on, bad stigmas around genetically modified foods because, in the end, they're good for us. You know, they, have effects that are the same as regular corn. You don't see the pigs gaining, 80 pounds a day, or you don't see the quail losing its fertility because they're eating BT corn. It's basically the same. Here's my references. Thank you guys so much for listening, and I enjoyed making this presentation. I enjoyed studying BT corn, and I'm definitely more informed than I was before, and I will forever be grateful for genetically modified foods because of, you know, there's so many possibilities that can be used. This is a great idea, and I think that as time goes on, genetically modified foods are going to continue to get better and continue. And in the end, I think that genetically modified foods can actually surpass natural foods in the fact that they they can be used for our own good and our own health. Like I said earlier, um, today 93% of corn is genetically modified in some way in the U.S. So even though people think that just because the label doesn't say genetically modified, you know, most of those foods and crops are, because those farmers want to be able to produce more, and without that, those toxins will get to them, those molds start growing, and it really hinders that growth of those crops. So yeah, that's my presentation. Thank you guys so much for listening, and thank you.